Good morning. My name is Clint Allen. I'm the senior author for the manuscript recently published in Science Translational Medicine entitled "The Tumor Microenvironment State Associates with Response to HPV Therapeutic Vaccination in Patients with Respiratory Papillomatosis." I'm an otolaryngologist here at the NIH, and Dr. Norberg, who's the first author of the work, is the medical oncologist who I collaborated with on this project. So we've been studying recurrent respiratory papillomatosis for a number of years now. RRP is a really devastating disease for patients that have this diagnosis. Even though it's not a malignancy, the recurrent nature of the papillomas and the standard of care treatment requiring frequent surgical intervention really destroys patients' quality of life, prevents effective voice and breathing, and it's a real problem. The cause of RRP is chronic infection with HPV 6 or 11, and so really new treatments designed to address the underlying cause of the disorder are needed. And so this publication describes our collaboration here at the NIH with Precigen, which is a biotech company here in Maryland, to develop a therapeutic vaccine designed to activate a T-cell HPV-specific response against an existing infection. And this work describes the phase 1 study of this novel therapeutic vaccine. What we observe is really an outstanding safety profile with primarily just grade 1 injection site reaction and mild prodromal symptoms, rounding out the toxicity profile. And in the recommended phase 2 dose, which was the second, the highest dose level used in the study, we observed that half of all treated patients required no clinically indicated interventions in the 12 months after the clinical trial compared to requiring interventions before the clinical trial. This was the primary outcome measure from a clinical standpoint that we, along with the FDA, has agreed upon as being clinically meaningful moving forward for these studies. So that response rate is really an early signal, albeit in a phase 1 study, that a significant proportion of patients really benefit clinically from therapeutic vaccination. Importantly, we did extensive correlative analyses to show that indeed the vaccine appears to have a mechanism that it should, i.e. that after vaccination, patients had greater HPV-specific T-cell responses in their blood compared to before vaccination. But importantly, what seemed to distinguish responders from non-responders was based upon the state of the papilloma microenvironment. Specifically, papillomas that had relatively higher HPV gene expression appeared to also have higher production of chemokines that recruit neutrophils into the papilloma microenvironment. And conversely, patients that responded very well to the vaccine clinically appeared to have low HPV gene expression and greater expression of the T-cell chemokines CXCL9, 10, and 11, and greater associated T-cell infiltration into papillomas. So it appears that HPV drives in different patients what we're considering to be a chemokine switch that really programs the papilloma microenvironment to be either susceptible or resistant to HPV-specific T-cells coming in from the periphery and doing their job. So this finding is spurring new lines of research in our lab and others to understand these mechanisms where HPV seems to be polarizing the microenvironment, with the ultimate goal of being able to manipulate this polarization to try to turn as many people as possible into responders to T-cell based immunotherapy such as this therapeutic vaccine.